Our first lecture in the series on target organ toxicity will focus on the liver. Hundreds of drugs or toxins can cause liver injury. The damage they cause can vary from elevations detected from liver enzymes to severe liver failure. The liver is the largest internal organ, and most xenobiotics or foreign molecules enter the liver via the gastrointestinal tract and the portal vein. It has a high concentration of metabolizing enzymes such as the cytochrome P450 enzymes that can be seen in the diagram in the top right. Many of the reactions that occur produce reactive metabolites, and the types of injuries that occur are not completely separate and distinct, meaning that one type can often progress to another. As is illustrated in the diagram on the bottom right, with hepatitis, cirrhosis, and liver carcinoma interacting with each other. The liver is also 2 to 3% of the body weight, and it is well supplied with blood vessels as shown in the diagram on the right, and it receives up to 25% of the cardiac output, which makes it particularly vulnerable to injury. The lobule shown on the bottom right of the diagram is a structural hexagonal unit, and it can be thought of as the functional units of the liver. It can be broken up into different zones containing many different cell types, Zone 1 being the closest to the portal vein, while Zone 3 is closer to the central vein, as shown in the diagram. Also visible are sinusoids, which are channels between the cords of hepatocytes, along with the endothelial cells which line the blood vessels, the Kupffer cells, which are like macrophages, and the Ito cells, which are fat-storing cells that respond to liver injury. Bile is produced in the liver and contains bile acids, amongst other things like glutathione, phospholipids, cholesterol, bilirubin, proteins, sometimes metals, and organic anions. Canaliculi, shown in the diagram on the bottom right, form channels between hepatocytes, and they merge into what we call the bile duct, which can be seen in the diagram on the top right. Bile is concentrated and stored in the gallbladder, and it is released into the duodenum, as can be seen in the diagram on the top. Lipophilic cationic toxins can pass in and out of these channels by transporter proteins shown in the diagram at the bottom right, such as the multi-drug resistant proteins like P-glycoprotein. There are many risk factors associated with liver injury. These include age, where there is often decreased clearance that can occur with age, more drug-drug interactions, reduced hepatic blood flow, poor diet, infections, and often liver disease. Alcohol can lead to the induction of liver injury, cirrhosis, inflammation, things like that, and cause a reduction in glutathione. This can also alter the metabolic enzyme activity in the liver. Genetic differences in DNA sequences coding for the cytochrome P450 enzymes can result in abnormal reactions to drugs and toxins. Concomitant liver disease, like that can occur with patients who have hepatitis or viral infections, cirrhosis, liver cancer, things like that, are at greater risk to toxicity due to drugs. Weight can often be a factor also, and body fat percentage can affect the distribution of some drugs. We will now discuss some of the mechanisms of toxicity that can occur in the liver. Hepatocytes, as we've already said, have high levels of enzymes, metabolic enzymes, in particular phase 1 enzymes, that can convert xenobiotics or foreign molecules to highly reactive metabolites. So concomitant intake of many different toxins can enhance individual drug toxicities. Non-covalent interactions, like those involving lipid peroxidation, can affect cell membrane integrity. Generation of reactive oxygen species can also induce necrosis or apoptosis. Depletion of glutathione, which is one of the major antioxidants in the cells, reduces the cell's ability then to scavenge and remove reactive oxygen species. Modification of individual chemical groups, like sulfur groups, for example, can reduce enzyme activity. And finally, covalent interactions with macromolecules like DNA can result in genetic mutation, which could lead to cancer. Cell death in the liver can affect other cells and the interactions that occur. Necrosis of hepatocytes results in leakage of cytosolic enzymes such as alanine aminotransferase or ALT into the blood supply, which can then be measured from a blood sample to assess the extent of damage. There can be different forms of cell death in the liver, such as necrosis and apoptosis. The differences between these are highlighted in the diagram on the bottom right. Necrosis involves the cell bursting open and the cell contents are leaking everywhere. This results in neighbouring cell damage and inflammation. Apoptosis, on the other hand, is characterized by cell shrinkage, nuclear fragmentation, the formation of what are called apoptotic bodies, which can be seen in the diagram on the bottom right, and no inflammation, as distinct from the inflammation that's often seen with necrosis. The extent of damage can also differ between toxicants, as shown in the diagram in the top right. Focal cell death, as it's called, involves randomly distributed death of single hepatocytes. Zonal death involves hepatocyte death in different functional regions, whereas pan-acinar cell death involves massive death with few viable cells remaining. In addition to causing cell death, many chemicals may cause the migration to the liver of inflammatory cells. Cell damage may result from the release of protease enzymes and reactive oxygen species. And therefore, with repeated exposure to certain chemicals, it may result in the development of an immune response, and in some cases, hypersensitivity reactions. Hepatic protein adducts, which simply just means the addition of protein groups on top of each other, can become antigenic and stimulate antibody production and activation of the complement system. Kupffer cells, which we said already are like the liver's macrophages, and Ito cells become activated after toxicant exposure. Activated Kupffer cells can produce cytokines and cytotoxins, and also reactive oxygen species and reactive nitrogen species. Ito cells, which respond to liver injury, secrete collagen. This results in scarring, which can ultimately develop into fibrosis. As was said already, sinusoids are channels between hepatocytes that carry blood throughout the liver. These are shown in the diagram on the right. Damage can occur by blockade of its lumen or by destruction of its endothelial cell wall, and blockade can occur when red blood cells can get caught in the sinusoids, for example, following large doses of paracetamol. Sinusoidal blockade results in the liver becoming engorged in blood, and this ultimately can lead to shock, which means a huge loss of blood pressure. Progressive destruction of endothelial cells lead to further loss of barrier integrity, which could then damage surrounding tissues and structures like the bile duct. Bile duct damage itself can manifest itself in many different forms. A particular form known as cholangiodestructive cholestasis is where there is damage to the intrahepatic bile ducts. This leads to serum levels of bile salts and bilirubin becoming elevated. And this is caused by swollen biliary epithelial cells and the debris of damaged cells getting into the lumen. Examples of toxins that can cause such damage include methylene dianiline, or MDA, which is an epoxy resin found in things like glue. Chronic toxin administration can lead to biliary proliferation and fibrosis of the bile duct, similar to biliary cirrhosis. And in rare cases, toxicity can even lead to bile duct loss, as can be seen in the diagram on the right. This is known as vanishing bile duct syndrome. Fatty liver disease is a reversible condition, and it occurs where there are large deposits of triglyceride fat accumulated in cells, otherwise known as steatosis. This is shown in the diagram on the right, where large white deposits of fat can be seen within cells. It is often associated with excess alcohol intake. As shown in the equation on the bottom right, which we've encountered already in the toxicokinetics lecture series, if you have more alcohol being introduced into the equation, it can lead to more NADH being formed and lead to an increased ratio of NADH to NAD. This leads to a decrease in the synthesis of lipoproteins. Toxins causing fatty liver include environmental hepatotoxins such as toxic mushrooms, carbon tetrachloride, which is a dry cleaning agent, chloroform, and also other hydrocarbons as well. Hepatic fibrosis or scarring results from the accumulation of extensive amounts of collagen fibers. At a macro level, this can be seen on the right in the comparison to a healthy liver. At a microscopic level, this is caused by scarring due to collagen deposition, as can be seen on the diagram in the bottom right. When it comes to cell division and these scars subdivide, the remaining liver mass results in nodules of regenerating hepatocytes. Chronic liver disease leads to the progressive replacement then of these liver cells with fibrotic scar tissue, and ultimately a condition called cirrhosis. Most commonly, this is caused by exposure to hepatitis or to alcoholism. Also, things like carbon tetrachloride and vinyl chloride can lead to it with chronic exposure. It leads to a reduced ability to detoxify xenobiotics or foreign molecules, cellular waste. It means there's a reduction in the production of bile. There's a loss of the ability to metabolize drugs, to produce plasma proteins, and unfortunately cirrhosis is not reversible and often leads to the requirement for liver transplantation. Hepatitis or liver inflammation is an acute injury to the liver due to the presence of inflammatory cells and can be acute or chronic. It can be caused by viral infections such as hepatitis virus A, B, and C, or due to toxins, or due to side effects of some drugs. Alcoholic hepatitis is a syndrome of jaundice and can lead to liver failure after decades of heavy alcohol use. At a macro level, you can see the difference here in a liver undergoing hepatitis and a healthy liver. At a cellular level, there are swollen hepatocytes that contain eosinophilic inclusion bodies surrounded by neutrophils or immune cells. Ethanol promotes the translocation of lipopolysaccharide or LPS derived from bacteria from the lumen into the portal vein, and this can cause liver inflammation. Liver cancer or hepatocellular carcinoma can occur secondary to hepatitis or to cirrhosis. A picture from an autopsy is visible here on the right that's suffering from liver cancer. About 30% of the hepatocellular carcinoma cases are related to excessive alcohol use. Chronic alcohol use for more than 10 years increases the risk of hepatocellular carcinoma 5-fold. It can also be caused by other products such as aflatoxin. Aflatoxin is a hepatic carcinogen and a byproduct of fungal contamination of foodstuffs, like that shown on the bottom right of the diagram. Aflatoxin causes DNA damage and mutations in particular of the p53 gene, which is known as a tumor suppressor gene. Humans are exposed to aflatoxin through the ingestion of moldy foods found in particular in susceptible grains.